For decades, the Y chromosome, one of the two human sex chromosomes, has been notoriously challenging for the genomics community to sequence due to the complexity of its structure. Just a few years ago, half of the human Y chromosome was missing the challenging, complex satellite areas, said Monika Sichova, co-lead author on the paper and postdoctoral scholar in biomolecular engineering at UCSC. The methods established in the paper will allow scientists to complete more end-to-end reads of human Y chromosomes to get a better understanding of how this genetic material affects the diverse human population. It was the Y chromosome that lacked the most sequences from the previous reference genome, said Arang Ri, a staff scientist at the National Human Genome Research Institute and the paper's lead author. Understanding rates of change on the Y chromosome and how to interpret this change are intriguing questions that will now be possible to study using the techniques developed in this paper to completely sequence human Y chromosomes. The richer reference that includes the full sequence of the Y chromosome satellite DNA will also allow scientists to better understand the evolutionary relationship of these sequences with satellite DNA found elsewhere on the genome. It is exciting to be able to finally see these sequences in heterochromatic, densely packed regions for the first time. But, because large parts of the human Y chromosome were missing from the past human reference, scientists were not able to identify them as human and thus mistook them to be part of the DNA of the species they were studying. This paper finds evidence that about 5,000 bacterial genomes in a common database likely contained contamination matching human Y sequences. People were guessing at it, but no one could prove that this was happening until now. While the complete human Y chromosome will open the door to many new discoveries, the researchers plan to further improve the study of this region by including the Y chromosome in future versions of the human pangenome. This will help scientists understand how the Y chromosome varies among people of different ancestral backgrounds and provide a better point of reference for understanding the Y across the diversity of the human population. The researchers hope to be able to collaborate with scientists around the world to enable others to complete Y chromosome sequencing. We aim to make these data widely accessible, Miga said.